Welcome to This Month in Type 1 Research. Please check the description for the original sources, and all content is general in nature and should not be considered medical advice. So, what if I told you that having a family history of type 2 diabetes might actually protect your child from developing type 1?
I mean, it sounds totally backwards.
Right, completely. Or, that the secret to preventing diabetic eye disease might actually be hiding in the blood vessels of the brain. Today, we are basically throwing out the old textbooks.
Yeah, we really are. We are looking at a diagnostic and therapeutic landscape that is completely shattering our traditional assumptions. For the longest time, type 1 diabetes, or T1D, was treated strictly as this mechanical failure.
Right, like it just happened to you out of nowhere.
Exactly. Your pancreas just quit, you took insulin, and well, that was the whole story.
And that is exactly why we are here today. We're unpacking a massive stack of recent peer-reviewed publications from PubMed, specifically from March 2026, focused entirely on type 1.
It's a huge stack.
It is. And whether you are catching up on the field for your work, or maybe you're living with T1D yourself, or you just, love medical science, our mission today is to cut through all that dense academic jargon.
You want to get to the good stuff.
Right. We are going to reveal the fascinating, highly personalized future of T1D care.
And if we lay this whole stack of research out, a really clear narrative starts to emerge. We're going to move chronologically through the disease timeline today.
Makes sense. Start at the beginning.
Yeah, looking at how the field is redefining onset, predicting, and actually delaying the disease.
Yeah.
Then we'll look at the daily reality for those living with it, specifically the algorithms taking over management.
It's the AI stuff, which is wild.
Oh, it's incredible. From there, we dive into some truly surprising biological discoveries involving gut microbiomes and neurovascular systems. And finally, we'll land on what might be the most crucial piece of all.
Which is?
The human and psychosocial realities of living with this condition.
Okay, well, we have to start at the very beginning of the timeline because the way we view T1D onset has just fundamentally changed. It's no longer viewed as this sudden, unpredictable event where you just wake up one day in the ICU.
No, not at all. It is now understood as a stage progression that we can actually intercept.
Intercept. I love that word for this.
It's totally accurate. And that paradigm shift changes the entire trajectory of patient care. We now formally stage T1D.
Walk us through those stages.
Sure. So stage 1 is having multiple islet autoantibodies in your blood, but your blood sugar is completely normal.
So the immune system has, drawn its weapons, but no damage is done yet.
Exactly. Then stage 2 is having those same antibodies plus abnormal blood sugar, but you still don't feel sick.
You're asymptomatic.
Right. And then stage 3 is the clinical diagnosis, symptoms hit, and insulin is required. Because we know these silent stages exist, the medical community is making a massive push for general population screening.
Which brings us to the ELSA study out of the UK. They're testing the feasibility of screening children in the general population for those early-stage autoantibodies.
Because historically, we only screened kids who had a parent or sibling with T1D.
Right, but most people who get type 1 have no family history at all.
Exactly. So the ELSA study highlights this incredible logistical hurdle of shifting from targeted screening to population screening. Finding those autoantibodies in the general population is, it's literally like finding a needle in a haystack.
Because you were talking about rolling out finger prick blood tests or venous draws to hundreds of thousands of asymptomatic kids.
Hundreds of thousands. You have to handle the physical logistics of shipping dried blood spot cards, analyzing them accurately, and this is perhaps the hardest part, managing the psychological impact.
Right. Telling parents their perfectly healthy 6-year-old is statistically guaranteed to develop a chronic autoimmune disease in the next decade.
It's heavy.
The psychological weight of that is immense. But knowing a storm is coming allows you to board up the windows, right? Let's talk about teplizumab, the therapy that takes advantage of that early screening.
Yeah, this is a huge development. It actually delays the progression to clinical stage 3 T1D by almost 3 years.
3 years.
3 years is a massive developmental window for a child. I mean, 3 years without needing daily insulin injections, without the risk of severe hypoglycemia, it fundamentally changes their childhood.
So how does it work?
Well, teplizumab is a monoclonal antibody that targets the CD3 TCR signaling pathway.
Okay, let's unpack that mechanism because CD3 TCR signaling pathway is a mouthful.
It is a bit much.
I picture this as like a peace treaty negotiation for the immune system. You have these reactive CD8 positive T cells that are acting like aggressive generals, launching attacks on the insulin-producing beta cells in the pancreas.
That's a great way to look at it. The T cell has a receptor on its surface. The TCR and CD3 is the signaling mechanism attached to it. Teplizumab binds to that CD3 complex and acts as a partial agonist. It sends a confusing partial signal that essentially overstimulates those aggressive CD8-positive T cells until they become functionally exhausted.
It was just tire out.
They tire out and deactivate, and simultaneously, the drug induces regulatory T cells, or TREGs, which act as the immune system's peacekeepers, restoring self-tolerance.
So you send in the diplomats to exhaust the aggressive generals and then bring in the peacekeepers to maintain order.
Exactly.
But let me push back on this for a second. The data shows this buys us about 3 years. If we can exhaust those attacking cells and bring in peacekeepers, why does the treaty inevitably break? Like, why can't we stop the autoimmune attack forever?
That is the million-dollar question. The treaty breaks because T1D is incredibly heterogeneous. It's not just a single biological pathway failing.
It's more complicated than that.
Much more. It is a highly complex dialogue between an individual's genetic susceptibility, various environmental triggers, and deep immune dysregulation. Exhausting a specific subset of T cells isn't a permanent systemic reset.
So the immune system just finds another way.
Yeah. Eventually, the broader immune system bypasses that specific roadblock. The underlying autoimmune drive finds a workaround. We are successfully buying time, but the root cause is still simmering underneath.
Which brings up the sheer complexity of the genetics involved. And this leads to, honestly, one of the most counterintuitive findings in our stack of papers today.
The TEDdy study.
Yes, the International TEDdy study, which tracks the environmental determinants of diabetes in the young.
Right. So the TEDdy study looked at children who are already in stage 1 or 2.
Meaning they have islet autoimmunity.
Yes, their immune systems have started the process. The researchers tracked their family medical histories to see what factors accelerated or delayed the progression to stage 3.
And what did they find?
They found that if these high-risk children have a family history of type 2 diabetes, it actually appears to significantly delay their development of clinical type 1 diabetes.
Wait, I want to make sure I'm hearing this correctly. Having a family history of a completely different metabolic condition, type 2, which is heavily associated with insulin resistance and lifestyle factors, somehow slows down the purely autoimmune destruction in type 1.
I know, it sounds crazy. But the researchers hypothesized that the genetic factors predisposing a family to type 2 diabetes might involve pathways that make the pancreatic beta cells themselves more resilient to stress.
Oh, interesting.
Because type 2 diabetes involves a long period where beta cells pump out massive amounts of insulin to overcome resistance before they finally fail.
They get a workout.
Exactly.
Yeah.
So while the type 1 autoimmune system is launching its attack, the beta cells of kids with a type 2 family history might just be structurally tougher.
They're built for endurance.
Right. They might be better equipped to survive the inflammatory onslaught, which stretches out the timeline before symptoms actually hit.
Wow. Okay, but even with that built-in resilience or, a 3-year delay from a drug like teplizumab, the underlying autoimmune drive eventually finds a way.
It does.
So what happens on day 1096 when the pancreas finally tips over the edge into clinical stage 3, that is where we have to abandon the biological delay tactics and hand the reins over to engineering.
Because once clinical onset occurs, the patient inherits a massive cognitive burden. I mean, they are forced to manually manage a highly dynamic biological system.
Which is exhausting.
But the integration of artificial intelligence is rewriting that daily reality. We have multiple scoping reviews and randomized trials in our stack looking at automated insulin delivery, or AID systems.
Systems like the FCL at Home trial or the Access Aid study in New Zealand?
Yeah, these are the algorithms taking real-time data from a continuous glucose monitor and automatically adjusting insulin delivery via a pump.
And the AI here is continuously learning the patient's specific metabolic patterns, right?
Yes, it uses constrained predictive control algorithms. Every few minutes, it calculates where your blood sugar will be in 30 minutes based on your current trajectory, the active insulin already in your body, and your historical data.
Okay, let me ask a grounded, slightly skeptical question on behalf of the listener here. We hear so much hype about AI these days, usually involving language models hallucinating facts or making weird logical errors.
Sure, I get that fear.
Is a machine learning algorithm really safe and nuanced enough to take the wheel and act as a human pancreas? What happens when it inevitably glitches?
It's a fair question, but these are not generative language models making creative guesses. They are highly constrained, mathematically bounded control algorithms trained purely on physiological data.
So they can't just make things up.
No. Safety parameters are hard-coded into the core.
Sure.
If the algorithm loses sensor data, or if the predictions hit a mathematically unsafe threshold, the system immediately defaults to a safe pre-programmed basal rate.
It has a failsafe.
Exactly.
Hmm.
And clinical trials consistently show that these systems dramatically increase the time patients spend in their target glucose range, specifically overnight, which has historically been the most terrifying time for severe low blood sugars.
And a vital piece of that automated ecosystem is the continuous glucose monitor, the CGM. And our sources show CGM usage becoming critical not just for kids or young tech adopters, but increasingly for older adults living with T1D.
Which is fantastic because for decades, the gold standard for measuring diabetes management was the HbA1c test.
Which gives a 3-month average of your blood sugar levels.
Right. But an average completely hides the dangerous extremes.
Yeah, HbA1c is like looking at your bank account's average balance over a year. It might say you average $5,000, which looks totally healthy on paper.
But that average completely hides the fact that you were overdrawn by $10,000 on Tuesday and won a $20,000 lottery on Wednesday.
Exactly. The CGM is your daily transaction history showing those terrifying overdrafts.
That perfectly captures the biological reality. A study in our stack by Stimson and colleagues found that CGM-derived metrics, specifically time in range and glucose variability, are a much stronger predictor of preventing microvascular complications like eye and kidney disease than the traditional HbA1c measurement.
Because it sees the spikes.
Yes. Because the CGM tracks the actual minute-by-minute fluctuations, it gives a true picture of the oxidative and metabolic stress the blood vessels are experiencing.
So technology is doing an incredible job of delivering insulin better and monitoring glucose better. But insulin doesn't fix underlying systemic metabolic dysfunction, especially as patients age or face modern health challenges.
That's a huge point.
So what happens when we look outside the traditional insulin-only docs?
Well, the dogma has always been that T1D patients only need insulin. But reality is far more complex. When individuals with T1D also struggle with excess weight or obesity, they develop insulin resistance, just like anyone else in the general population.
This creates a double burden.
Exactly. They lack insulin production, and their bodies resist the insulin they do inject. This brings us to the emerging use of GLP-1 receptor agonists and tirzepatide in T1D patients.
These are the medications making massive headlines globally for weight loss and type 2 diabetes management.
Right. And adding a GLP-1 or tirzepatide to a T1D protocol improves their glycemic control and drives significant weight loss.
Which is great.
It is, but most importantly, early data suggests it actively improves major cardiovascular and kidney outcomes, which are the primary drivers of mortality in older patients.
So if insulin is the fuel keeping the plane in the air, giving a T1D patient a GLP-1 is like adding a highly skilled co-pilot.
I like that.
It slows down digestion to match the absorption curve of the injected insulin, smoothing out post-meal glucose spikes, and helps navigate the turbulence of weight management.
And the co-pilot actively reduces the total amount of insulin the patient needs to inject, which reduces the sheer volume of exogenous hormones circulating in their system.
That's fascinating. And as we look deeper into the biology, the surprises just keep coming. Let's move from metabolic hormones to the gut microbiome.
Oh, this study was wild.
Yeah, we have a fascinating 16S RNA gene sequencing study on diabetic mice.
So genetic sequencing allows researchers to basically take a full roll call of every bacteria present in the gut. They induced type 1 diabetes in these mice and then treated them with regulatory T-cell therapy.
Those are the immune peacekeepers we discussed during the teplizumab segment.
Exactly.
Yeah.
Then they sequenced the gut bacteria to see what happened.
Wait, I need to make sure I understand this connection. We inject a therapy into the bloodstream to alter T cells, and the downstream effect is a completely different bacterial ecosystem in the digestive tract.
Yep.
How does a T cell in the blood even talk to bacteria in the intestines?
Well, the Treg cell therapy physically altered the diversity and composition of the mice's gut microbiota. Specifically, it lowered the ratio of firmicutes to bacteroidetes. In untreated diabetic mice, this ratio was extremely high.
Okay, but why do we care about the ratio of two specific types of bacteria? What does a high firmicutes ratio actually do to the body?
A high firmicutes ratio is typically associated with systemic inflammation and a compromised leaky gut barrier.
Oh, wow.
By bringing in the Treg peacekeepers, the gut environment shifted to favor bacteroidetes. These bacteria produce short-chain fatty acids like butyrate, which actually reinforce the gut lining and calm the immune system locally.
So it's all connected.
It proves there is a bidirectional street between our systemic immune system and our gut flora. Modifying the immune system directly reshapes the environment of the gut.
That implies that autoimmune regulation and the gut microbiome are deeply physically tied together in a continuous feedback loop.
They absolutely are.
So if we want to truly treat T1D in the future, we might need to actively culture the gut biome alongside modulating the immune system.
It forces us to view the immune system as an ecosystem, rather than just an army we need to suppress.
My mind is slightly blown by the gut connection, but let's move from the intestines up to the brain.
Let's do it.
There's a longitudinal study on the Akita mouse model, which is a genetic model of type 1 diabetes. The researchers look specifically at neurovascular coupling.
Neurovascular coupling is the vital mechanism where blood flow increases in the brain in response to neuronal activity. When a specific part of your brain works hard, say you're solving a math problem, it demands more oxygen and glucose.
Right, it needs fuel.
Specialized cells signal the local blood vessels to dilate and deliver that fuel.
And we know that in diabetic retinopathy, the eye disease common in long-term diabetes, this vascular coupling gets severely damaged.
Yes.
The blood vessels become brittle and stop responding properly to the demands of the tissue.
And the historical assumption has been that if the tiny blood vessels in the retina are failing, the tiny blood vessels in the brain must suffer the same severe dysfunction.
Which would make sense.
But this study found that neurovascular coupling is surprisingly preserved in the cerebral cortex of diabetic mice, even as the disease progresses for months. The blood vessels in the brain still dilated perfectly in response to stimulation.
That is incredible news. It means the brain has some sort of intrinsic protective mechanism against diabetic vascular damage that the eyes and the kidneys simply do not possess.
Yes.
If we can figure out exactly how the brain is shielding its blood vessels from the oxidative stress of high blood sugar, maybe we can synthesize that mechanism and apply it to the rest of the body.
It completely challenges our assumptions about how systemic diabetes complications really are. The body's tissues react to metabolic stress in vastly different ways.
Wow. Okay, well, we have spent a lot of time talking about AI algorithms predicting glucose, peacekeeper T cells altering gut bacteria, and the brain protecting its blood vessels. But you pointed out in our outline that all these biological and technological advancements mean absolutely nothing if we ignore the psychosocial realities of the human beings living with this condition.
Yeah, we have to look at the epidemiological data, and it provides a very sobering reality check. There is a 14-year retrospective study out of Auckland, New Zealand, looking at community deaths from diabetic ketoacidosis, or DKA.
DKA is the acute complication where the body lacks sufficient insulin to process glucose, so it starts burning fat at a rapid rate, releasing ketones and turning the blood acidic.
It's awful.
It's an intense, life-threatening emergency, and people usually associate it with newly diagnosed children who haven't started insulin yet.
Right. But the demographics in this Auckland study tell a completely different tragic story. The median age of death from DKA in the community was 55.
55 years old.
And the most striking statistic, 58% of these individuals lived alone.
Wait, 58% live completely alone?
Yes. The major factors driving these fatalities weren't a lack of access to the newest algorithmic pump technology or novel adjunct therapies.
Then what was it?
The fatal factors were social isolation, socioeconomic deprivation, inconsistent medication routines, and a total lack of clinic attendance. These individuals were simply disengaged from the healthcare system.
Let me challenge the entire medical focus of our earlier sections based on that fact alone. We can engineer the most advanced AI pumps and develop miraculous stem cell therapies in the world. But if 58% of people dying from DKA are 55 years old, living alone, and disconnected from care because of burnout or poverty, aren't we failing at basic community support?
We are. It highlights a massive, fatal blind spot in modern medicine. We focus so heavily on molecular targets and device engineering that we ignore the heavy, daily cognitive and emotional burden of managing a chronic illness.
The day-to-day reality.
If a patient is deeply isolated, dealing with depression, or struggling to afford rent, the most sophisticated AI algorithm cannot save them if they lack the bandwidth to engage with their care.
That ties directly into a qualitative review in our stack regarding how a T1D diagnosis impacts an individual's core identity.
The review explored the concept of biographical disruption. A diagnosis forces a person to entirely rewrite who they are.
How so?
Well, they have to integrate this relentless, demanding disease into their sense of self. It affects their personal relationships, their ability to be spontaneous, and their fundamental trust in their own body.
That makes total sense.
If clinicians don't acknowledge and treat that identity disruption, we lose the patient to burnout.
Which is why the Promise workshop paper is so refreshing. Researchers brought together adults with T1D and specifically asked them to define what matters to them regarding future stem cell-derived islet therapies.
It is the ultimate example of patient-centered development. A scientist in a lab might think the singular goal of a stem cell cure is achieving a mathematically perfect HbA1c graph.
Just flatlining the data in a good way.
Right. But the researchers realized that to a patient, a cure means something entirely different. A cure means being able to eat a slice of pizza at 8 p.m. without doing complex calculus to figure out if the fat and protein ratio will crash their blood sugar at 2 a.m.
They want their mental bandwidth back.
Exactly. They want freedom from continuous device alarms and the background terror of severe hypoglycemia.
So if a new stem cell therapy requires heavy daily immunosuppression that makes the patient constantly feel sick or fatigued, even if their blood sugar is absolutely perfect.
To the patient, that is not a successful therapy. They are trading one massive cognitive burden for another.
They want their identity and their freedom back, not just perfectly functioning beta cells.
Yes. And when you view the disease through that lens, you realize that successful treatment has to alleviate the mental burden just as much as the metabolic one.
To summarize this incredibly deep dive, type 1 diabetes is rapidly transforming. It's evolving from a strictly managed, unpredictable chronic illness into a highly personalized condition.
It really is.
We are learning how to predict its onset with population screening, delay it with immune therapies that alter our very gut biome, and automate the grueling daily management with highly constrained predictive AI.
But we cannot lose sight of the fact that looking at health from multiple angles, from microscopic cellular resilience to the deep need for community social support, is the only way to actually save lives. And, the pace of this innovation leaves us with a fascinating prospect to consider.
Let's hear it.
If future therapies can successfully delay the disease by decades, and targeted AI algorithms can completely automate whatever management remains,
Yeah.
will the very identity of what it means to be a diabetic fundamentally shift?
Wow.
Will it evolve from a defining, all-consuming chronic illness into a quiet, background biological quirk?
That is a profound thought to leave on. Remember to like and subscribe, follow the link in the channel description to buy me a coffee, and thank you for listening.